Hello everyone, I welcome all of you to my channel. This is MEENU AGRAWAL. Now moving with the topic further, this is your Microsporogenesis. Okay, we will deal with the entire topic leftover, seed formation, all those things we will look at in this video, and I will try to end this chapter in this video. Okay, so moving on further, as you can see here, this is the female part of your flower. This is your stigma, style and ovary. And here this is your what? This is your carpels are fused together. You can see here, and this is an example of your what? Of your syncarpus. Syncarpus. No? And this is an example of your carpel. So this is a syncarpus carpel. And this is your what? This is your apocarpus. Ah, fine. Now you can see here that this is what? This is a your ovule. It's showing the structure. Okay. Now if you look at here, what is funicle? Funicle is where is the stalk. Funicle is the stalk which is joining with your what? It is joining the ovule with the placenta. Okay. So just let me help you to write it. So funicle is what? Funicle, you can say it's a stalk, that helps in joining your ovule. It joins your ovule with the placenta. Okay, with placenta. Okay, so this is your funicle, and the second is your hilum. You can see it's what? It's a junction. It's a junction between your funicle and your ovule. So it's a junction, you can say, now between funicle and your ovule. Okay, now if you look at the ovule, this region which you see is your what? Is your micropyle, and at this region there is no integument is present. Okay, now you can see that it has got two layers of integument, that is your outer and inner integument. Okay, and just inside it is your nucellus. Okay, so this part I can show you. This is your nucellus. Okay, so this part, this region is your nucellus. Basically this is the region which is providing the nourishment. It provides what? Nourishment. Okay, so you know that the nucellus provides nourishment, provides nourishment to the embryo sac. Embryo sac. Okay, so it provides the nourishment to the embryo sac, and this is your embryo sac. Okay, and fine. So the end opposite to the micropylar end is known as chelazal pole. Okay, so with this we will move on, and we will see now how your megasporogenesis is happening. Okay, now coming back to this slide, as you can see, this is your cell, now which is your nucellus cell only. And remember, nucellus cells only act as what? They act as your microspore or a megaspore mother cell. They act as your what? Megaspore mother cell. Okay, they act as your megaspore mother cell. So one of the cells of the nucellus, as you can see here, it becomes what? It becomes quite enlarged. Okay, and it's filled with cytoplasm and all, and it will undergo, now see, it is written also megaspore mother cell, and it undergoes meiotic division. Okay, it will undergo meiotic division, and you can see at it's giving rise to 4 megaspore tetrad. It will form more megaspores. And remember one thing, again I am reiterating it, this nucellus cell, this megaspore mother cell is 2n, and now what is happening is, this is your meiosis 1 will happen, so 2 cells will be formed, and then meiosis 2 will happen. So Meiosis, don't forget these things. Meiosis 1 gives rise to 2 cells, and then meiosis 2 will give rise to 4 cells. Okay, and now these each cells are what? N. They are all having n number of chromosomes. Now, out of that, the 3 degenerates. What happened to them? The 3 degenerates. The 3 degenerates. Okay, 3 degenerates. On only one of them will further start grow. So only one of them will further start grow. Okay. So now what is happening? If I just rub this one. Now what will happen? 3 will degenerate and only one will further move on. One will further go on into what? Mitosis. You can see here the cell will enlarge and it will into mitosis. This is mitosis 1. As a result of mitosis 1, what has been formed? What can you see here? Due to mitosis 1, 2 nuclei has been formed. Okay, they are n. Okay, they have n number of chromosomes. They have n number of chromosomes, and here also they will have. It's going under mitosis. Therefore, it will have n number of chromosomes. Okay, these nuclei. Now this is mitosis 1 and this is mitosis 2. Okay, so how many nuclei you get? 4. Okay, then this is mitosis 3, and what will happen? 8 nuclei will be forming. Okay, so what you see here, 8 nuclei are formed. Okay, now in the embryo sac, what changes do you find? You find that the 3 nuclei moves to one pole, 3 moves to another pole. Okay, and the one at the micropylar end, you will find always egg at the micropylar end. Okay, so you will find that the 3 cells are moving at the micropylar end and 3 move at the chalazal end, and these cells are called as antipodal, and they will form what cells? Okay, they will form membrane around them. Okay, so you will find that these are what? Antipodal cells. Okay, and these cells are called as what? At the micropolar end, 3 cells will be formed. One is your egg, as you can see here in the diagram. Okay, so one is your egg, as you can see here. This is your egg, the cell, and these 2 cells are your synergids. Okay, and these are your what? These are your polar nuclei, and this bigger cell. See this bigger cell in the center, which you can see. Okay, which you can see is your what? The center cell is your central cell. So there are how many cells? 3 cells here, 2 synergids and 1 egg cell. Okay, and 3 antipodal cells and 1 central cell which is having 2 polar nuclei. Okay, so in and all, how many cells do you see in this embryo sac? You will see in this embryo sac that it has got 8 nuclei. It has got 8 nuclei, and it has got how many cells? It has got 8 nuclei and 7 cells. Okay, this is the characteristic feature, remember, of your embryo sac of an angiosperm. Okay, and we will see how the pollination is going to happen. Okay, so now I will just draw for you this embryo sac for you. Now this embryo sac is there. Okay, and just I will draw this part also of the pistil so that the things become more clearer, and I will just make it bigger, try to make it bigger for you people. Okay, now what do you find? That this is your embryo sac. Okay, now this is suppose your egg cell, and these are the 2 synergids. Okay, now these are your antipodal cells. Rather, I should use this color for your synergids. Okay, and egg cell is white. Okay, these are your 2 synergids, and then I will make 2 polar nuclei. I will make in the center 2 polar nuclei. Okay, and you can see here I'm just making the nucleus. in each of them, not visible. Okay, so you can see here, right? So similarly you have to also practice of making these diagrams. Okay, now you, this is your pollen grain which is falling on it. Okay, you can see pollen grains are there. Now remember that your these now what happened that these uh, your synergids basically they now release a chemical, okay, which is a small protein which is produced by the synergids in the ovule. Okay, they produce a small protein. Okay, they produce a small protein which is producing synergids, and they produce and they guide. And I have already told you that there is a constant dialogue going on, and if the this pollen is of different having gene, then due to self incompatibility they don't allow it to grow, and synergids release a small protein, okay, which helps in guiding this pollen tube to reach to the egg. Even if I destroy one of the synergids, if you destroy, okay, then also the pollen tube can really reach. But it has been seen experimentally, if both the synergids are destroyed, then the pollen tube cannot be guided to the egg. So there is a constant dialogue, chemical dialogue going on, because the synergids release a small protein. Okay, now with this what will happen, ki this now when this pollen tube comes, now let me make another diagram for you people. Okay, so this is your embryo sac. Okay, now when this pollen tube is coming, rather I should do like this. This is your embryo sac. I have to highlight them, therefore I am doing it for you. Now this is your what, um, these are your antipodal cells. Okay, three antipodal cells are there. Okay, uh, अच्छा तो नहीं बन पाया, but still I will try. Three antipodal cells are there, and in the center you can see this is your egg, and it has got two synergids. Okay, so it has got two synergids, and synergids, you know, they have a filiform apparatus and a non-like structure. You can draw it like this, and they have a nucleus, all haploid nucleus, you know, and in the center you have a cell which is called as what, which is your central cell. Okay, so I'm drawing it for you. This is your central cell having two polar nuclei. Now this pollen tube which comes, it has got what, you have read about microsporogenesis also, so you know about it, that they have got two male gametes on them. Okay, so if it is not visible, then I will use another color. They have two male gametes. Okay, so they have these two male gametes. Okay, now if you look at these male gametes, what will happen, they will come and they will fuse, these pollen tube will go inside, and one of the male gamete will go and fuse with the egg. It will be directed and it will fuse with the egg. Okay, and one of the other male gamete will go and it will fuse with your polar nuclei. So one will fuse with the egg and other will fuse with the polar nuclei. So what do you get here, you get now here a zygote will be there, which is having what, which has got fuse, fertilization has taken place, now it has become 2n, and what you are going to get, you are going to get the central cell, है ना, I'm just showing you two polar nuclei, and if I suppose that this one Male was yellow. Okay, with a different color I am drawing it so that things become clear. Okay, so and these will be antipodal cells here. Okay. Okay. Now these three will fuse together, polar nuclei and your male gamete will fuse together and it will form your what? They will fuse together and they will all NN, they will fuse together and they will form 3N, which is your endosperm. Okay. And what will happen to the rest of them? With passage of time, your synergids and all, you will see they will just wither away, and the zygote will start growing. Okay. Now let's move on further and see how the zygote grows. Now you can see the structure. Okay. Now you can see the degenerating synergids, as I have told you. They start degenerating, and here also the antipodal cells, they start degenerating. Now you see here what is your this is your zygote, as you can see here, and this is your what? Zygote. Okay, this is your zygote. Now there are two things inside that embryo sac. One is your what? One is your zygote and the other is your what? It's your endosperm. Now remember that this endosperm is a nutritive tissue. It plays a nutritive, it's a nutritive tissue. It's a nutritive tissue. So it will help in what? In feeding the developing zygote. It will help in growing the embryo. It will feed. It will help. It will provide nutrition to the developing embryo. So which should divide first? In endosperm should divide first, or your endosperm should divide first, or your zygote should divide first? What should divide first? So definitely your endosperm will divide first. So you will see that endosperm divides first. Now which kind of division will take? Mitosis. It divides first, and remember this kind of fertilization which we have studied, because your male gamete is once fusing with your what? With your egg and giving rise to male gamete, plus X gives you what? Zygote, which is 2N. Okay, and male gamete plus your and this polar nuclei fuses and it gives you what? Endosperm, which is what? Which is 3N. Now this kind of fertilization is called as what? Double fertilization. What do we call it? Double fertilization, which is unique. Which is unique to angiosperms. This is a unique character. Okay, this is unique feature, and you find them only in angiosperms, not in any other group, neither in gymnosperms, nowhere. Okay, so this is called double fertilization because one male gamete fuses with the egg, another fuses with your polar nuclei. Okay, and endosperm is your nutritive tissue which will divide first, so that some nutrition is available for the growing embryo. Embryo will require nutrition, so this endosperm will divide first. Now let's look at this diagram. Is not very clear, and remember I have told you that zygote is present towards the micropylar end. Now just I will show you here in this diagram. Just look at it. What do you see here? What do you see here? That just see. What do you see here? You see here that this is the micropylar end. This is the micropyle, and this is the area where your zygote is formed. Now this zygote divides, okay, and it forms your, firstly it divides and it forms your suspensor cells. Okay, and these suspensor cells, they this bigger cell, can you see, this is called as oustorium, which helps in deriving nutrition from this endosperm. This yellow part is what? This yellow part which you see is your endosperm. Okay, so you can see this, and this is your endosperm. Okay, so just highlight it. This is your endosperm, as you can see here. This is your endosperm. Okay, yellow part. So it has divided first, and then your zygote has divided and it has formed the suspensor cell, and this part is your embryo part. Okay, this stage is called as what? Pro-embryo stage. This is the earlier stage of the embryo, and then after this comes your next stage. Okay, after this comes your globular stage, which is the next stage. Okay, in this you will find that more division has taken. Okay, and it has taken the form of a ball of cells. Okay, which is called as globular stage, and then comes the stage which is called as heart shaped stage. Okay, this stage is called as heart shaped stage. Just look at it, it is forming the heart shape like structure. Okay, and further you get what? You will see that the stage is called as torpedo stage. What do we call this stage as? We call it as torpedo stage. Okay, the stage is called as torpedo stage because you can see here that the cotyledons starts to be formed, and cotyledon, and slowly what you will see that totally the endosperm will get used up. Okay, just see, I'm just showing you this diagram, just look at it. You can see here the endosperm is decreasing, isn't it? Endosperm is decreasing because it is getting used up as the embryo is growing. Okay, and you can see here, this is your what? This part is your what? It's your apical stem, apical meristems are there. Meristematic cells, you know, they are what? Totipotent cells. They are undifferentiated cells. We have talked about it earlier, and they can differentiate and can give rise to various other parts of the plant. Okay, and you can see here. Okay, and here you can see is what? It's your root apical meristem. So this is your shoot apical meristem, and here you will find is your root apical meristems, and these are the two cotyledons which are containing nutrition for the development of your seed later, and your embryo is getting used up. Okay, so depending upon whether the endosperm is there or not, the further seeds are being categorized. Okay, and you can see that this your outer integuments which was there in your ovule wall is now changing into what? Into seed coat. Okay, so outer wall is called as testa of the seed. Okay, the seed coat they are forming. Okay, so the outer one which we call as what? The outer one which we call as testa, which is hard, and the inner we call as tegmen. This is what we call them. Okay, and these are the two cotyledons, as you can see here. Okay, they will further develop, and these are the different parts of an embryo. Okay, this is called as what? Plumule, and this is called as what? This is called as radicle, which will give rise to your what? Root, entire root it will give rise to, and plumule will give rise to your what? Shoot. Okay, and based on whether the endosperm is there or not, your seeds are divided into two types. What are they? They are your endospermic seed. They have endosperm. They have endosperm, or we also call them as albuminous seeds. Okay, they have endosperm, and the other is non-endospermic seed. Okay, so whether they have or not, and mostly you will find that the monocots have got endospermic seed and dicots have got non-endospermic seed. But we will also see that there are certain cases where it's a dicot plant but it is having non-endospermic seed. Okay, so let's look on the this is there. Okay, now you can see here, now this is your now seed, which is a dicot seed, basically a kidney bean. As you can see here, this is your kidney bean and this is your corn. Okay, corn everybody eats, भुट्टा सब कोई खाते हैं, इसलिए आपको पता है. So you can see the outermost covering of that, now your integument has changed into what, into your seed coat. So if you look at the parts of the flower, now we will come to that also. We will see now those petals and all sepals and all, they will wither away. Okay, and what will change, your now ovary, your ovary will change into what, your ovary will change into fruit, isn't it? And the outer ovary wall, okay, it will form the fruit wall. Okay, so ovary wall will form the fruit wall, which is called as pericarp. Okay, we will look at these structures. And then your ovule, ovule changes into what, into your seed. And you can see on the seeds, you must have seen there is a opening, a dot like structure you must have seen on the seeds, which is your micropyle, through which your exchange of gases and all happens. So that is the micropyle region. Okay, so this is your ovule changes into seed, and you can see this integuments over the seeds, they changes into and into your seed coat. Already I have told you about testa and integument, and then the seed coats changes into pine. And then if nucellus, you know that nucellus provides nutrition to embryo cell, and in some cases you find that there is nucellus, persistent nucellus, mostly it not degenerates, but in some case if it is persistent, then it forms your what, perisperm, and it is seen in case of your black pepper. Okay, so that is there. Okay, in the seeds we find them nucellus in some cases, not every one of them. Okay, so these are the some changes that takes place in your integuments, nucellus. Anything else is there? Okay, so we will move on. Okay, now cotyledons, you know. Okay, moving on further. Now here you can see in the picture that what is it they is showing you. It is showing you what? It is showing you, this is your monocot seed, and this is your what? Which kind of seed? It is an endospermic seed. It is an endospermic seed. Mixed seed. Why we are calling it as an endospermic? Because it has got, you can see it's an endosperm. Okay, and you can see here this is written as scutellum. Here the cotyledon is known as, this is scutellum, or you can say cotyledon. Some new terms students see, they get what is this now. So this is your scutellum. Okay, now in it you will only see that they have got a covering. Okay, your plumule is covered by a structure which is known as coleoptile. We call it as what? Coleoptile. Okay, this is covering your what? Plumule. Okay, plumule meristems are covered by your coleoptile, and your radical meristems are covered by coleorhiza. Coleorhiza. Okay, so these they are a protective covering which protects your meristems. Okay, and you can see here you can see that the ovary and now ovary wall is your basically your pericarp. So now you can see here it's quite now close together to the seed. Okay, now this one you can see this is your what, this is your dicot. Now castor seed, this is your, this one is your castor seed, this is your castor seed. Okay, now what do you see with this castor seed, this is your dicot. As you can see, this is an exceptional case which is having what, it is having, you can see here it is having endosperm. Okay, so it is having endosperm, so it is an endospermic seed, but it is a dicot. Okay, so from here we just see ki there are various examples which is present there. Okay, so and here you can see in this maize plant you can see here that this is your called as alluron layer. Okay, this is called as alluron layer which is surrounding your endosperm, this is called as alluron layer. Okay, so these are the different uh, parts of your what, seed. Okay, now we have seen the different parts of the seed and now we will move to the fruit. Okay, now this diagram is very important, as you can see here in this diagram. See, first I will focus here. Okay, and on this diagram you can see this is your pentacle. Okay, stalk by of the flower. Okay, now you can see here that these are the sepals. Can you relate it well? Because here is the persistent sepals are there. Okay, this is calyx, persistent. No? Sepals are also collectively called calyx. So persistent calyx is here seen in case of tomato, not in like in brinjal also it is seen. Okay, so here this is there, and what inside you see these are the patterns, they all wither away, they are not there anymore. Okay, now you can see inside this part which is your ovary. Now related, here the ovary has grown. Okay, this is the ovary which is grown. Okay, and this is the central part which is your placenta. Okay, to which your what ovules are attached. Okay, now here your seeds are attached, you can see here the seeds are attached. Okay, so it's called as what, it's a true fruit. Why? Because here the ovary is developing into a fruit, so such kind of fruit is called as what, it's a true fruit. We call them as what, we call them as true fruit, because here ovary is changing into fruit. Okay, so it's a true fruit, and you can see style and all they go away. Okay, now coming to this is an example of your false fruit. Why I'm saying false fruit, just now take your attention here to this part of the, ना, just you see here that there are sepals, आपको ये कभी कभी दिख जाते हैं ना एप्पल में आपको, और एप्पल है ना, आपको ये नाशपाती है जो हम लोग कहते हैं. Okay, so what do you see here, here you see that the, now this is the locule and this is the seed, isn't it, and this is the part of the ovary. Okay, this is the part of the ovary. Now what has happened here, here there's this floral tube. Can you see this floral tube, this part of the thalamus, okay, has grown here. Okay, so the part of the thalamus has grown here. You can see this is your floral tube and this is the part of the thalamus. Okay, now this has grown fleshy here, so this part is your floral tube, which is your thalamus. Basically the part that you eat is your thalamus, it's not of your what, ovary. You can see here ovary is this part and it is restricted to this part. You can see here, if it can make out, जो हम लोग निकाल देते हैं seed के साथ जो part होता है थोड़ा सा कटा हुआ, that is basically your ovary. Okay, so you can see here in this diagram, and this is your seed. So basically the edible part is your thalamus, it's not your ovary, so it is called as what, it is called as this is an example of your what, false fruit. So this is an example of your false fruit. So you should be clear okay, what is false fruit and what is a true fruit. Okay, fine. So moving further uh, with this, and now we are coming to another part, that is your parthenocarpy. Now, in case of parthenocarpy, what do we call parthenocarpy? Parthenocarpy or parthenocarpic fruits are what? They are seedless fruits. Fruits formed without fertilization. Okay, they are called as your parthenocarpic fruits, and there are certain factors which result into parthenocarpic fruits, like there are genetical factors, okay, which induce it. There is certain environmental factors like your frost, fog, all these. In certain cases it induces in certain plants, okay, parthenocarpy. And there is certain now hormones like auxin. No, we can like farmers, they use auxin to induce parthenocarpic fruits. What is an advantage of parthenocarpic fruit? That you get rid of your the seeds, rid of seeds. You get rid the seeds. When it comes into mouth you feel irritated, na? So you get rid of seeds, and it's also good for the industry which is taking out fruit juices. It's good for them, but for plants it's of no value. Why? Because plants, they try to spread themselves by with this formation of seeds, okay. So if you look in that way, so yes, no pollinators are required in this case. No pollinators are required, you know? So these are the features. And but basically if you look at from plants perspective, it is not beneficial to them because they don't form, no seeds are there, so no seeds. So it's not good for them. For they can't disperse themselves by this. They use seeds for the dispersal, for propagating themselves, so which is not happening in this case, okay. Now there is another term which we call as apomixis. Now in case of apomixis, what is happening now here? Now it is the formation of seeds. Now here the seeds are formed without fertilization, without you can say without fertilization, or you can say it's an asexual mode of seed production, or you can, a better term will be asexual mode of seed production. And I have told you the seeds, they use it for the dispersal. So they have these seeds, you know, it's a way of asexual mode. You can see, which is basically promoting seed production and which is helping in the dispersal. Mainly in them, what do you find that the nucellus cells, now they now lead into the formation of your seeds. So no, now basically there is no fusion of gametes taking place. Huh? This is an asexual process. Huh? And embryo has developed from an diploid cell. This is what? This is your 2N cell. This is your diploid cell rather than from a diploid zygote. Okay, so this is what? This is a diploid cell which is giving rise to your seeds rather from a zygote. No zygote is formed in this case. No zygote is formed. Okay, no fusion of gametes is taking place. So it's an asexual mode of reproduction. Why we are talking of apomictic seeds? Because it is very good for those hybrid plants. If I have a hybrid plant and by this asexual method if I get seeds, then it is very easy now to grow those plants, because there won't be segregation of characters. Okay, because by sexual method segregation of characters happen. Okay, so by asexual method that won't be happening, and therefore now genetics the people in this industry, they are working about how we can get these apomictic seeds, and these will be very good for those. We won't need to grow our hybrid high yielding variety seeds. We need to form again and again, but we need to hybridize them again and again and then get hybrid seeds. But in this case it's not the thing. Okay, so that's why it will be very helpful for this industry. Now with this we have finished our chapter sexual reproduction in flowers. So please go through it. If anybody has any problem, please comment it in my comment box. You can ask me. I will be very happy to answer you people. And with this I will say goodbye and God bless you.